So we're in our last little section here, and we're going to try to do justice to liverworts. I realize that I'm probably being biased. I'm being biased or something because I emphasize mosses because that's what I'm interested in. But my graduate students that work on liverworts would say, you know, why didn't you start with liverworts? You should describe them first, and I probably should have. So we're going to give liverworts equal time, equal exposure here. Liverworts are a little bit more complicated because they have so much variation. If the phylogeny is right and the division between liverworts and the rest of the land plants is the earliest, then the liverworts have had the longest time to evolve on their own track and develop different things. I don't know if anybody knows this one. This one's up in the chaparral right now, or it ought to be in any normal year. This is Asterella. It's one of our beautiful little liverworts, which for some reason I relate to because it has this white beard of scales that hang down below this structure I'm going to talk about in a minute. So if you see a liverwort that has these structures with the white scales here, then you're dealing with Asterella. We'll talk about what exactly those are. That looks like a sporophyte, and it isn't a sporophyte. It's actually an extension of the gametophyte. So this stalk is part of the mother gametophyte, and so is the umbrella. And the gametangia are born underneath. And then the capsules develop hanging down there. You can see some capsules right here that are brown. So that's the capsules. These kind of liverworts do a different thing than even other liverworts do or than mosses do. They're not using the sporophyte itself to raise up into the airstream. They're raising a part of their gametophyte up into the airstream, and they get these little umbrellas, and then they're releasing the spores from underneath them. So we'll look at that a little bit, but I don't know if anybody recognizes one of these, but this is something you'd see under shrubs in the chaparral or on banks in the East Bay Hills all the time. It's a wonderful little group. I have a graduate student that's doing PhD work on this. So let's talk about liverworts, and they really have a much more diversity than mosses do in their basic structures. There's basically two big different morphologies. There's the thalloid ones we talked about before. That's where there's a flat sheet of tissue instead of stem and leaves. And there's even two types of thalloid liverworts, which we'll go over, the Metzgeriales and the Marcantiales, two big orders of liverworts. But they all have that flat sheet of tissue, and some of them tend to have those raised up umbrellas. The umbrellas, this is Marcantia, which is one you might know. This grows in gardens and greenhouses in California. What is going to be the umbrella, initially you'd say, like, well, how does it ever get fertilized being up there? Well, it starts out down here. So that's going to be an umbrella someday. And it does its fertilization here. And then the stalk that's going to bring it up is going to happen later. The characteristics of the thalloid liverwort is that strap-shaped plant body. And the thallus is variously organized. I'm going to talk about that in a minute. Oil bodies are this unique organelle that's only present in all of life. It's only present in the liverworts. And they are either in all the cells or they're in specialized cells. So they may not be in all of them. The spores are, the whole seta and sporangium is very ephemeral, a bit more like a mushroom, where their lifespan of that phase is basically overnight and then releases its spores and it's over, where unlike the mosses, where that process can happen over days or weeks. But the two types, so one type is called the complex thalloids. And this is a characteristic one here, conocephalum. This translates in the Greek, cone means cone, and cephalum means head. So it literally means the cone heads. And those of us that grew up in the Saturday Night Live in the 1970s, they had the skit, the cone heads. So easy to remember. These have what looks like a little cone-shaped sporophyte. And Conocephalum also has the really, really checkered top of the, and I'm going to say what those little dots are, but it looks like an alligator skin. This is one that also occurs commonly in California. It occurs outside of our academic building on campus. I can usually go out and get some for classes. It likes stream banks and underneath rose bushes if there's not a lot of litter. So you probably don't have too much trouble finding this. And this is one that has a chemical cue to it. You can smell this from 20 feet away. It has flavonoids that are really nice smelling. So if you take a little bit of this and crush it, it just smells great. So in Sobe Free, we always advertise like, come and smell the liverworts. So Conocephalum, really fun. It's just an example. That Asterella is another complex thalloid. And the reason we call it complex is because of its anatomy. This is a cross section. So if we took the thallus and just cut it in a thin slice, we'd see that there's a lot of structure to it. It has an epidermis. It has a green layer, a spongy layer of photosynthetic cells. It has big storage kind of cells below that, and it has an epidermis on the bottom. Anybody recognize what that looks like? Like, what could that be of things you're familiar with? Anybody say? Anybody ever looked at a cross section of an angiosperm leaf? It's remarkably like this. So this is not a leaf like anything that an angiosperm would have in terms of evolution. It's a parallel evolution, not homologous, but it has the same kind of structure. These openings here are not stomates, but they're pores, P-O-R-E. They serve the same function as a stomate does in a leaf, except that they don't close. So these, we look at the alligator skin, look at the top of the liverwort. Each of those white dots is one of these pores. I always say, you know, the Linda Ronstadt sang a song about this in Poor, Poor, Pitiful Me. So these are the pores, and they're actually slightly homohydric. They're maintaining a higher water content in here. They're not fully poikilohydric, actually. So they are doing a little bit like a angiosperm does. And then they have a division of labor where the photosynthetic cells are in filaments. The palisade layer in a leaf, if you remember your leaf anatomy, in angiosperm leaves, there's a layer that's spongy photosynthetic tissue with air spaces between it, which is analogous to this. It's so that gas exchange can happen more easily with the environment. And then you have these storage cells where photosynthetic products are stored in the storage cells, parenchyma. So they have they have 5 or more morphological types of tissues building in the thallus. So that's what we call them, complex thalloids. And all the ones, many like Marcantia, which you might easily know, and even the Asterella, those are all complex thalloids. You see the pores on the surface of the Asterella here, and there's even pores on the surface of the Archegonia 4, which is that raised up portion. So they're pretty easy to tell. The other kind of thalloid form is the simple thalloid, and they're called simple not to denigrate them, but just because they don't have tissue differentiation. They only have a 1 or 2 cell thick thallus, and the cells are all the same. So this is a very typical looking one. It looks different than the complex thalloids. And phylogenetically, I'll show you in a little bit, it is different. It's not directly related to the others. Some of them look almost leafy. This is an extremely common liverwort in the California flora. It should be all over the place in the hills now. It is thalloid, but it has leafy lobes that make it look a little bit leafy. They're just frills. I call it the Mexican wedding shirt liverwort. If you think about formal dress wear in Mexico, you have these shirts that down the middle, you have these kind of frilly additions. So that's what's going on here. They're not really leaves, but they're kind of fancy lobes of the thallus. And these black olives that are sticking up here is what you would see about this time of year in March. Those are the capsules. And the seta is just this little watery stalk, very unlike any moss. Those are mosses for contrast in between here. There's actually two different mosses. There's this one and this one. This one's probably Timiella, you can tell by, that's one that has two strata in the leaves. And this could well be Ceratodon, which I was making fun of earlier. So you can find this all over the place. And I put this picture in just to show you, this is a highly magnified view of a cell of either a simple thalloid or a leafy liverwort. They're the same. The chloroplasts, which are a common organelle in plants, that's what these are. They're just chloroplasts. But these weird looking things that look like a bundle of grapes, those are the oil bodies. And in fresh liverworts, you have extremely distinctive oil bodies, and some of the best characters to tell fine taxonomic divisions within liverworts would be the form of these oil bodies and the color of them. These are grape-like botryoidal ones, but some might be round, and some might be football shaped and so forth. So when we talk about oil bodies, that's what we're talking about here. It's those of you who know cell biology, the endoplasmic reticulum, which is a series of membranes that's inside the cell. It's thought that this is derived from that, kind of a specialized version of that as food storage. So that's kind of interesting. So we have Fossembronia and Pelleas and Metzgeria. This is Anura, which we have in our flora by water. You can see some mosses sort of straggling in there for contrast. So that's a moss. But this maybe even looks to you a little bit like a complex thalloid, but if you were to make a cross section of it, you'd see it. It does not have this epidermis and photosynthetic layer and storage layer and so forth. This Metzgeria has a little bit of complication because it has a midrib down the thallus. So I guess it's not entirely simple. This is the midrib right here. This is a cross section of that. And most of the thallus is a single cell thick, and then it has multiply cell thick right in that midrib. And you can probably guess what this is. That's the seta, and that's the sporophyte, the capsule. And it's doing something weird. We'll talk about in a minute how they open. They don't have a peristome, and they don't open like moss ones do. And these branches, you might not recognize what those are down there. Those would be male branches. So bryophytes either have the male and female on the same plant, or they have them on different plants, a little bit like angiosperms, except in angiosperms, we call it dioecious and monoecious. In bryophytes, we call it dioicous and monoicous because we're talking about distribution of sexes that are on the gametophyte. Whereas in angiosperms, we're talking about distribution of sexes on the sporophyte, the diploid. So there's genetic differences between, genetic consequences of having separate sexes on the gametophyte versus sporophyte. We can talk about that more in the discussion section. People wonder, why do biologists call things monoicous instead of monoecious? That's why. So here's a simple thalloid. One other thing it illustrates here that I didn't emphasize too much, I pointed out the rhizoids and the mosses. The rhizoids and the mosses are multicellular extensions of the vegetative body. In liverworts, they're unicellular. There's no cross walls in there. So these have a little bit more of the morphology of a root hair that ferns and angiosperms would have. They're just a unicellular extension, whereas the mosses are more complex. They have cross walls and they're much more elaborate. And then the largest diversity actually of liverworts are leafy, but they're leafy in a totally different way than mosses are. These are the ones that would confuse you perhaps with mosses when you're not very familiar with bryophytes. But as I mentioned, when we were talking about mosses, they tend to have the leaves arranged all the way around the stem. And liverworts fundamentally are sort of dorsoventrally symmetric, so they're flattened. The This would be a typical liverwort here. That's a leafy liverwort. That in this case, it looks like something weird's going on. There's lobes of these leaves that are folded under the stem. And there are sometimes under leaves, which are separate leaves. We can't really tell the difference between the under leaves and the lobes here. But this would be one of these guys this is Frulania, which is one of our really common liverworts that grows on trees around here. You can find it in Berkeley Hills. It has a very characteristic color, this kind of deep burgundy color. It's desiccation tolerant, but it has a look to it. See how much flatter it is than the mosses we were looking at. So in general, if you see a really flat bryophyte looking plant, it's a liverwort, unless it's the few of the mosses like Physidens that are also flattened. But there's a couple of things that also tell us it's a liverwort. There's no midrib here. So there aren't midribs in liverworts. And they tend to be bilobed. Many of them are bilobed, which no moss is. The two lobes in this liverwort, though, they're asymmetric. That's one lobe, and that's another lobe. And the lobe is kind of folded together. If you think about the kind of mitten design that humans wear, where we have 4 fingers in one big part of the mitten, and we have the thumb in the other part, and the thumb kind of comes up and opposes the rest of it. That's what's going on here. It's like a mitten where the thumb would be this lobe and the big part would be the big part of the mitten. So that's a taxonomic character that you would use, would be the kind of lobe and how it's folded. Does it make a little sac, or is it folded flat? Things like that. The Frulania actually have a little sac arrangement to them. So they generally appear 2 or 3 ranked and dorsoventrally flattened, no midrib, rather simple construction as compared to the mosses. They can be a little bit hard to tell. I mean, the big, robust, leafy liverworts can look a lot like a moss, but you see how this one is still pretty flattened? And with a little bit of practice, you could tell this from a moss. Here's another one, quite flattened, even though it has pretty big leaves on it for a bryophyte. Here's one where you see these teethed leaves. We have 2 different ways that the leaves are arranged. They're either arranged on in such a way that the leading edge, if we're looking at the top of the liverwort, the leading edge of the leaf below is on top of the leaf above it, or else it's arranged the other way where the leading edge of the leaf below is underneath the leaf in the front. And those have some very interesting names that come from mythology, which I'll just throw out there and you guys can look it up and see. I'm not going to go into the details of it, but the one arrangement is called incubus and the other arrangement is succubus. And those have meanings from Greek mythology, which you should look up. We can write that in the chat. I don't want to go over it in mixed company. So the this is kind of the characters we're looking at for liverwort identification. Is we're going to look at the leaf arrangement, and we're going to look at, this is one of the succubus ones where the this edge, this is the top of the stem here, this edge is overlapping this one. Whereas if it was the other way around, it would be incubus. So we're looking at the leaf arrangement, the leaf cell shape, the oil body shape, the underleaves, color, and the microscope preps are really simple for these. We don't dissect away the leaf like you do in a moss. To look at it under the microscope, you just slap the whole thing down on a slide and put a cover glass on it, and you can go right ahead with key. You got to pay attention to which is the upside of the liverwort and which is the downside. Okay, so they are leafy, usually dorsoventrally flattened. They're simpler leaves than the moss. They're 1 cell thick, but no midrib. There are oil bodies, which are those some kind of strange color or shape that vary quite a bit in the cells. And one trouble with collecting them is those oil bodies tend to break down after the liverwort is dry for a while, especially if it wasn't very desiccation tolerant. So what most people that study liverworts do is they photograph the oil bodies right away, or else you just put your collection in the refrigerator for a little while and don't let it dry until you're ready to take pictures of it, so you can see. To get these beautiful oil bodies like this, to get a picture like that that shows those, it has to be fresh material. It's one trait that doesn't show up well from herbarium specimens. Okay, so here's just some random variation. Here is a close-up of the leaves. These are a leaf cell, and all the way around the edge of the leaf cell lined up is the chloroplasts. We can tell they're the chloroplasts because they're green, and that's where the chlorophyll is, and that's where photosynthesis is happening within the cell. And then these weird structures here are the oil bodies. This has a different kind of oil body than the one I showed you before, but they have a different refraction to them because they really are full of oil. They're food storage places. So it's pretty easy to tell them, especially with fresh material. And the shape and size and number of those is really important in the keys for telling fine scale, like species within a genus sort of level. So we have, it's hard to tell from this picture which is overlapping which, but we need to pay attention to how the leaves are overlapping. And the leaves are sometimes greatly divided. Look at this, where the leaf is divided into filaments so much that it almost looks like filaments. But this has 3 filaments, I believe, in each leaf. So it is it's so serrate that the filaments are almost, more than everything else put together. So you'd be able to key that out with filamentous leaves. Check this out. This is Frulania, what it would look like up close. That's this one here. If you wet it and then turned it over and looked at its underside, remember I mentioned that it has inflated little lobules. These little structures here are actually part of the upper leaf. Like if you think of the mitten analogy, that's like the thumb of the mitten. But in the case of Frulania and that whole family, Frulaniaceae, the lobe is inflated. It's like a little sac. Literally, you can see an air bubble in that one. And they act as little apartment houses for those rotifers. So if you get a fresh Frulania and you look at it, don't be surprised if you saw rotifers in that KQED film. Don't be surprised if you saw one rotifer in each of these little sacs. It's kind of its home. It's like an apartment building for rotifers, each of them eating away sort of freely. And it's probably a water storage device as far as the liverwort is concerned. It really does hold water in a little packet there. So that's kind of cool. The big tropical radiation of leafy liverworts is in the Frulaniaceae, which is this group, and there's a few of them in temperate areas. And then the Leguniaceae, which is a relative group that also has underlobes, and they have thousands of species in the tropics, really poorly known. They're one of the groups that's radiated onto angiosperms as a substrate. Here's the sporophyte. I got a picture coming up that's probably a little bit better. Let me see if this works better. I first want to show you the 4th type. The 4th growth form are these bottle liverworts, spherocarpus, which is on an evolutionary branch pretty much by itself. You can see these really easily in the Bay Area, up in all the East Bay parks, very often under picnic tables in places where the lawn is not thick. If you look right about now, you'll see in this cryptobiotic crust, you'll see these bottle-looking like green sacs. Each one of those has one gametangium inside. So there's one female gametangium in each of those little bags, and each of those, if it gets fertilized, will then produce a sporophyte. And the males are dimorphic, so the males also have sacs, but they're really small. So all of these purple colonies in here are the males. And so you can usually find, this is a dioicous species, and you can usually find the males and females growing together. If you know what a tetrad is, right after meiosis, sometimes the four products of meiosis hang together as a group and get dispersed as a group. That's called a tetrad. And in this case, probably those two males and those two females were part of the same tetrad, because you very often find two females and two males in proximity to each other. So these are really fun. And it's kind of an archaic group, not very diverse, and has some really bizarre structures to it, but it is actually quite abundant. It's an annual weed, basically, here in the Mediterranean climate sort of areas. So I don't know if anybody's ever seen this, but it's really a lot of fun when you do. Okay, so what happens with their sporophytes? So we have the seta that is not like the moss seed at all. It's very watery and thin and ephemeral, just lasts a day. And then it has this olive, so it's like an olive on a stick. There's no operculum. Remember, in the moss, we had a calyptra, then we had an operculum, which was an opening, and then inside of that, we had the peristome. We don't have any of this with the liverwort. We just have this olive, which typically breaks into four valves, just splits down the side and breaks open like this. There would be a few exceptions to that, but that's the general pattern, and it tends to be explosive. And by explosive, I mean that it happens all at once. If you were watching this under your stereoscope and it was getting heated by your lamps and you're watching it, you'll just see a little pop. It'll just go poof, and it'll pop open like this and just throw the spores out. So it does its spore dispersal as definitely one big event. It just kind of blasts open and blasts the spores. And helping that are some auxiliary structures that are not peristome, but they have a function a little bit like a peristome. So if we look up close, and this picture David Wagner gave to me, by the way, he's an excellent photographer, and he uses the photography stacking software to get really good depth of field. So this is one of the complex thalloids with its incredibly complex spores. These are individual haploid spores. And these spring-like looking cells that are mixed in with them are called elaters, E-L-A-T-E-R. When the capsule is closed, those are all condensed down like a spring that you've forced to be all condensed, but it wants to open, and so they're putting pressure. This elater is like a spring that's held in tension, and it really, really wants to release itself. And so while the plant's developing and while these are developing, they're building pressure, and then all of a sudden they just blast open the sporangium, and then they throw these spores all over the place. So we have no peristome, but we have elaters, and the elaters are not found in the mosses. So we have peristome in mosses, elaters in liverworts. And many of the really complex, difficult taxonomic groups, you actually use spore ornamentation to tell them apart. So a really good microscope obviously is pretty necessary for that. Rixia, for example, it's a good thing that you can look at the spores. So that's really different. This is what I was saying at the beginning of the lecture this morning, that if we want to compare and contrast mosses and liverworts, and hornworts, which we're going to talk about in a second, the sporophytes are more different from each other than the gametophytes are. And that's because each of these sporophytes has evolved completely independently of the others. Their common ancestor would have had a very, very simple, maybe just a sporangium, but no stalk at all. So the whole mechanism of elevating the sporophyte and opening it up and dispersing spores, there were separate solutions made in each of the bryophyte lineages. And by the way, you can see the remnants of the eliters here. Some of the eliters have one end stuck to the wall of the sporangium and the other end free. And so they're, really, you can see how that just loosens and blasts out that mass of spores. I love that picture. Okay. I don't know why I put it in the wrong order. Maybe I just skipped it. Oh, there it is. I knew I had a phylogeny in here. You couldn't get away without one phylogeny at least. Okay, so here's what we think the phylogeny of the liverworts are, and that also has been really well worked out. The phylogeny of liverworts and the phylogeny of mosses internally are quite consistent, and different data sets give the same result. So even though we're not 100% positive what the relationship of the different bryophyte groups are to each other within the two really diverse ones, liverworts and mosses, and also within hornworts, the phylogeny is really clear. So how do we read this thing? The mosses and hornworts and tracheophytes and the green algae are all outside here. So the liverworts are this lineage right here. Right here. There are a few of these archaic groups that branch off first. These are really interesting. They are thalloid, but they have leaf-like lobes to them. They look much more moss-like, these early diverging groups. For some reason, they've tended to survive more in the southern hemisphere. So if you go to New Zealand, you see an awful lot of diversity in quote, quote, primitive liverworts, or at least early diverging ones. And there's a big split above that group. There's a big split between the complex thalloids that we looked at and the simple thalloids plus leafy. So there's kind of a shocker, but it's well supported that the two groups of thalloid liverworts are not together a lineage, that the simple thalloids are actually more closely related to the leafy liverworts than they are to the complex thalloids. That's what this grouping right here tells us, which tells us that the complications that developed in the thallus of this group that includes Marcantia and Asterella and things happened later after it diverged from the main line here. And you can't, maybe not read it, but Sparocarpus, the bottle liverwort that I showed you, turns out to be in the complex thalloids, even though it doesn't have a very complex thallus, but it's an early diverging group within that clade. And all of these branches up here would be things like Conocephalum, Marcantia, and Asterella. And there's many others. It's Rixia, another group I showed you. It's really quite diverse, and this is the most desiccation tolerant, desert loving of the liverwort lineages. You can see some of these guys right out in the Mojave Desert. We'll see them on Subby-Free. Whereas most of the leafies are much more delicate and the simple thalloids are not quite as desiccation tolerant. So we have that shocker where the simple thalloids are closer to the leafies in an evolutionary sense. And then interestingly, there's two very distinct groups of simple thalloids that together they don't make up a monophyletic group. These ones like Fossembronia, the Mexican wedding shirt liverwort, are distinct from some of these other ones like Pallavicinia and the Pellia, some of these here. So that's kind of cool. And then there appears to be a single evolution of true leaves in the liverworts. And there's a couple of different major lineages of the leafy liverworts, which we won't go into detail, but one of them is the one that has the Frulania type of leaf and it's more tropical. And then there's some others that don't have that sort of mitten-like lobe to them. So it's really clear, you can tell from this, that the leaf of the liverwort evolved completely independently of the leaf of the moss. Because if we went back to that moss phylogeny, the leaves evolved in that clade and they have a different structure. They're not even very similar to these leaves. So leaves arose in these two places, leafy liverworts and mosses. And then leaves appear to have evolved at least 2 times in the tracheophytes. It's thought that the leaf that the lycophytes have is a different evolutionary origin of a leaf because it has a different anatomy than the megaphyll that's shared by ferns and seed plants. So there's at least 2 different times, and possibly a few more, where leaf-like structures evolved within the tracheophytes. So leaves are, one of these good ideas in evolution that seem to have been hit at in a bunch of lineages separately. It's the division of labor idea where you have a flat sheet of tissue that can be perpendicular to the sun's rays and capture the sun in a way that a kind of a spirally arranged set of stems can't do nearly as well. So this is liverworts. This is an even older group. The bottom of this tree is old, so 500 million years. Some of the very first fossils of land plants look like liverwort spores. So this is an archaic group in one sense, but as I was saying for the mosses, there are several places here where there has been recent evolutionary activity and origin of a lot of new lineages. So old groups can reinvent themselves. I guess it's a bit like the birds. So the birds are a remnant lineage of dinosaurs, just one lineage of dinosaurs, most of which went extinct, but then the birds themselves have undergone irradiation in more modern times. So the take-home message from that is just because a group is old and archaic doesn't mean that it doesn't have current evolutionary activity or it doesn't produce new lineages or something like that. And then I'm just going to talk about hornworts briefly, and then we'll have a chance for some more interplay. Hornworts are a much less diverse group. There's only a few hundred named species of them. So they possibly may be one of these groups that's a little bit more relictual, that has had a lot of extinction in its past. They have really distinct characteristics, the most obvious of which is the horn refers to the sporophyte is not distinguished into a seta and a capsule. It's kind of all capsule. So it's producing spores all the way along its length. And they're produced over a long period of time, which is not like the liverworts and mosses that produce all their spores in a given sporangium at once. They're not super photogenic, and they're not as morphologically variable. So, I don't know what you think, but they tend to be not quite as distinctive looking. But one thing they tend to have is, you see this blue-green color. We get lots of hornworts around here in our ephemeral California flora, and they tend to be very blue-green. And it's because they virtually all have particular blue-green bacteria, blue-green algae that live inside of them. They have little chambers that they make for their endosymbionts. And there's a regular relationship between the plant and its blue-green bacteria that it contains within it. It tends to have this kind of blue-green greasy look to it. My colleague Ken Kellman calls this the gummy bear look. So when you see the blue-green gummy bears, you're kind of guessing you got a hornwort instead of a liverwort. But once you see the capsule, you're pretty darn sure. So characteristics-wise, they really do look like thallose liverworts. But there are some major differences. They have these cavities inside them that have nostoc, which is a type of cyanobacteria that does have a role in nitrogen fixing and so forth. So the nostoc is a regular partner with these guys, almost as intimate as a lichen is with its algae. There is, for some weird reason, usually only one chloroplast per cell. Sometimes there's more, but I showed you that cell of a liverwort a while ago that had tons of chloroplasts around the margin of it. These, all the ones in our flora, just have one huge chloroplast that looks more like a dinner plate and that takes up the whole cell. Go figure. Don't know exactly why it does that. There, you can see mycorrhizae all the time in these. In liverworts and in hornworts, there's really clear mycorrhizal connections. seems to have been lost in mosses. You have the reproductive structures are not on any kind of fancy thing like in liverworts. They're actually in pits on the surface of the thallus. In an early picture at the beginning, I showed you the antheridia kind of down in little caves. We have the horn. There's stomata on it. So there's stomata on the sporophytes of hornworts and mosses, but not on liverworts. And the distinctive thing about hornworts as compared to both mosses and liverworts is that their spores are shed sequentially and continuously over an extended period of time that could be months, where a given capsule of a moss or liverwort pretty much does its thing all at once. Here's one more picture. This is showing what I'm talking about. These are the cells, and you see there's just one gigantic plate-like chloroplast in there. Just usually one, maybe two. So under the microscope, you just immediately, even a hand lens, you can actually see the chloroplast under a hand lens. And you go, oh, that's definitely a hornwort. This particular one, Dendroceros, you'll see that in the tropics. That's not a California thing, but as an epiphyte in the tropics, you have this weird looking hornwort. It actually has a midrib. And this just reminds me to say, this is found in liverworts and mosses too. Occasionally you get a kind of a precocious germination of the spore while it's still in the sporangium. And so the unit, when it actually disperses, this is the spore, but it's already had a bunch of cell divisions. So it has already the new generation kind of rearing to go. So that is, again, epiphytes tend to do that among the mosses and hornworts. All these Dendroceros are epiphytes. You tend to have a little bit of an advance of your germination, so they're kind of ready to go. And I didn't put in a hornwort phylogeny picture because they're pretty simple, but they're quite clearly distinct from the other two groups, and they have their own interests. There's one lab in Montreal that studies them. And there's even going to be a course, I think one of those Eagle Hill courses is going to be on hornworts. Hornworts usually get ignored. But if you look at that schedule Stacy put in there, I think there's going to be some hornwort discussion. They actually have a lot of interest to them. So here's the three groups again for review. Here's our hornwort. May not look like much, but this is what it looks like in about April. In our area, they look for all the world like blades of grass, and they have a basal membrane just like a grass leaf does. And they're producing spores, maturing spores as they go. And the top of it is the spores have already dispersed, and the valves are splitting open a little bit like a liverwort capsule. So the spores are coming out at the top, and they're still being matured in the middle, which is unique to the hornworts. This one is the genus Anthoceros. It has black spores, which you can kind of see. They're clinging to the valves of the sporangium. And our other common hornwort is Phaeoceros, which has yellow spores. That's what, so they're very easy to tell apart, even with a hand lens. One has yellow spores, one has black spores. So there's the hornworts with their unique structure. Here's our cone head liverwort again, Conocephalum, with its sporangia, with its watery seta and sporangium. And then here's our moss again, with its, this is one of the nodding mosses, one of the Bryophytes type of mosses with the pendant sporophytes. But I hope that by now you can at least tell the difference between the major groups of Bryophytes, and you know something about the kind of characters we use to separate them taxonomically and put them into groups.